Welcome to Halteres Presents, a brand-agnostic interview podcast that seeks to objectively highlight the happenings within the world of diagnostics. And now, your hosts, Thayer and Mickey Urdea.
Hello, this is Rich Thayer, Managing Partner at Halteres Associates.
And this is Mickey Urdea, the founding partner at Halteres Associates. Today we're interviewing Dave Engelthaler, who's the Director of Pathogen Genomics at TGen North in Arizona. Dave has had a long-term interest and involvement in both epidemiology and pathogen analysis for a variety of different infectious diseases involved in public health.
And today we're going to hear some fascinating thoughts on new genomics tools and how those tools are being used to survey a variety of outbreaks in the United States. In particular, I liked his response at the end, tying together all his thoughts and mentioning how AI is helping to advance use of these tools in the clinical sector. And now our interview with Dave Engelthaler.
Welcome to the podcast, Dave. Really a pleasure to have you with us.
It's good to be here, Mickey and Rich.
Welcome.
Thanks. So to get started, please tell our listeners a bit about yourself.
Yeah, sure. Happy to do so. So I'm Dave Engelthaler. I'm the director of the Pathogen and Microbiome Division of the nonprofit Translational Genomics Research Institute. My background is primarily in infectious disease epidemiology and then the application of new tools, especially genomic-based tools, to really advance that field. But maybe to go a little bit deeper on who I am, I'm really more, I see myself as a field biologist at heart. I grew up as a biologist. I got my undergrad at Northern Arizona University. Mickey, you know a little bit about that.
Yeah.
In wildlife biology, I was a field biologist for the Forest Service for years. And then there was an outbreak of a very unusual disease that occurred in the Four Corners area of the United States, which we subsequently found out to be caused by a hantavirus. It was hantavirus pulmonary syndrome. And that was right about the time I was doing field work, and public health needed, a biologist to go out and do habitat assessments and understand which rodents and which mice are carrying this and what parts of the country. And so I jumped into public health and I've never looked back. I've been part of public health for, just about 30 years now. And having worked at the state level, the federal level at Centers for Disease Control up in Fort Collins, Colorado, and having worked at the local level too, I really wanted to make sure when I moved over to where I'm at now, TGen, or the Translational Genomics Research Institute, that we brought public health as a really important thing that we translate to, not just to, better diagnostics for patients, other tools for clinical medicine, but also to population health and public health and One Health-based areas. So it's been really great to be in this translational research world, because I get to apply all the things that I used to do in the field or in the clinic or in behind the epidemiologist computer to this cutting edge science.
It's been a pleasure to watch all the work you've done over the last several years, which is why we were very excited to ask you to come to the show. So I know that you and your team have been advancing the use of NGS to investigate infectious diseases and other health issues as well. So what are some of the exciting advances in the technology, and how are they improving the use of NGS for your studies?
Yeah, that's great. NGS is it's really a game changer. Where we're seeing a lot of things happening with sequencing is smaller, faster, cheaper, more flexible, more accurate, and more useful, but really only if you want and know how to use it. So this is not just some kind of fancy technology that lab geeks run. This is now becoming more and more commonplace applied in the real world. As I mentioned, our goals are to see how do we move this into clinical medicine, but also into public health and into the One Health space, thinking about animals and the environment. So I think sequencing for sequencing's sake, just because it's out there, really probably has minimal value. But there's still value in that because you're generating new data around genomes of organisms of interest, and that can be added to the context of everything else that's out there for others to use. But if you know how to use sequencing to obtain what I like to refer to as pathogen intelligence, Now you have a lot of power. I mean, pathogen intelligence, at least the way that I look at this, is the knowledge that's gained from the information that we gather from genomic data, as well as surrounding metadata around samples. And so, you know, really thinking of this, to sequence a pathogen's genome is to reveal its secrets, to know what it's most related to, or what it's not related to, or what drugs it's resistant to, or will it be able to escape vaccine or previous infections? Does it have known virulence factors? Has it recombined with other pathogens like we see with influenza? All of that we get that intel from sequencing the genomes. And because the technology, as I mentioned, is getting smaller, faster, and cheaper, it's becoming more accessible. It's becoming more democratized, essentially, in a way, not just that more people can sequence, but more people can use the actual data, information, knowledge, and intel that comes from sequencing.
Mhm. Are you also able to use smaller or different samples, or can you see a broader set of organisms now than you could before? Or, what's changing, and is that helpful?
Yeah, let me use maybe an analogy here. May not be the best one, but I mean, I think that the next generation sequencing technologies that we're using now to develop this pathogen intelligence is really equivalent of the James Webb telescope to explore the universe. I mean, we think that previously, we could look up and see the stars without any real technology, like we can see people getting sick from disease, but we didn't have the resolution of information, other than the fact that maybe some stars were brighter than others and they'd seem to move position in the sky. But then along comes Galileo, who, in his telescope, and now we are really starting to get some level of resolution, down to the level that we could see planets, we could see moons moving around the planets, and start to understand that there's other galaxies. These are equivalent in the diagnostic world, in my mind, to some of the early diagnostic testing, where we could really detect and differentiate and understand different pathogenic microbes, and then do some testing, say, against certain drugs. But thinking back in the astronomy side, after several hundred years, we really advanced exponentially with the Hubble telescope, which is really the first time we're putting a telescope into space now to see, get past our atmosphere. Now we're seeing with much greater resolution, exponentially greater resolution. And now we're getting that intel on the universe that I was trying to talk about with pathogens. And we can explore galaxies and black holes and things that we really had to make a lot of inferences about before. This I think is probably similar to when we brought molecular testing on board into the clinic and into our labs with, especially PCR. We have superior accuracy over all previous testing than we could. We can look for genomic markers of specific phenotypes like drug resistance. We have, though, still a limited ability to only look for really what we're looking for. We can't go beyond the reaches of what the lens of the Hubble could do. But now with the James Webb telescope, I think this allows for us, well, it's absolutely allowing for us to gather, I think, real, cosmological intelligence. We have a higher resolution. We have can see deeper and further and earlier into the beginnings of the universe. And this, I think, is what pathogen intelligence is getting from NGS. I mean, we are now, I think we are really limited. It's pretty amazing. We're really limited only by where our, the limits of our imagination can go. We can see the first 2 cases of an outbreak with such high resolution. We can exclude things that aren't part of an outbreak. This is precision epidemiology, something we couldn't do before, but you have to know how to apply it to epidemiology to make that work. We can dive deep into minor populations of an overall infection, which is, a lot of times our infections are multiple different subpopulations, and we can see the very earliest beginnings of, say, resistance occurring in a TB patient, where a TB patient may get treated for months and years. We could see that resistance forming way before that patient fails therapy. So now we can actually help really develop precision clinical medicine for infectious disease, which we haven't really had. We've been really doing that for the last 10, 15 years in cancer, but now we can do that with infectious disease. And so there's a lot of things, we can monitor population changes in other long-term infected patients, like, even some immune suppressed COVID patients where these new variants would then pop out because they'd have been affecting a patient for a long period of time. We can monitor that evolution. We've been monitoring viral evolution in HIV now for a while. So we're really seeing precision medicine entering into the infectious disease space because of the appropriate applications of next generation technology.
That was a great analogy, by the way. Just loved it. And it reminded me, for some reason, of the quote, consternation becomes elucidation, which was from the Firesign Theater. Yeah, that's great. Thanks. Thanks very much.
I think maybe one other place to add to that too is, and I mentioned the kind of environmental and One Health space. There's a lot of areas. I think one, that's really interesting, and we're still exploring it and how best to use it is things like wastewater based epidemiology, which is I think it's becoming a critical tool for public health. But again, for those who understand both the capabilities and the limitations, and then what we can gather from that. But we can see not only the presence of specific pathogens, so like we look for COVID in wastewater, we can see the viral load or the pathogen load, so we can make some inferences about the prevalence in a population, and we can look at different sizes of population depending on where you're sampling from. But we can also understand things like population diversity. We can see the actual diversity that's evolving within a population, which does give us real information about how much transmission's occurring. That is actually critical information. Is it just one person? Is it one small population? Are we seeing a lot of different populations moving around? And we were able to see this with COVID would be occurring before, a couple weeks before we would see increases, say, into our hospitals. And then we could even actually look for very specific markers, such as monoclonal antibody resistance markers, things like that were really important during certain parts of the pandemic. We could see how that's going to be important for other organisms. And then I'm thinking also like emerging infections, or those things that that we're really concerned maybe coming into our community. With wastewater, we could have essentially like a bolo of be on the lookout for, things like mpox or polio. And in fact, we could find those in populations where we only know of maybe one case occurring. You could actually see that in a whole community's wastewater sample. So then that gives you an assessment that something is present and actually may be larger than what existing epidemiological and clinical surveillance tools provide us now. So I think we're just scratching the surface, but it is making a difference. But again, you have to kind of have a bit of the imagination as well as the knowledge base on capabilities and limitations, and then bringing this to the other parts of the One Health space, looking in the environment, in the air, water, soil, vector, animals, all those things that we think of as important things for us in public health, but we don't have good tools to monitor the pathogens that may be, or the pathogenic microbes that may move across that space into the human side. So I do think that we're really just starting to just see the horizon of where this dawn of the genomic age is for improving overall human health.
Yeah, we did a video on eDNA last year, which we're big fans of. Yeah,
fascinating.
Yeah, Dave, thank you for that fascinating discussion on the advancement in technologies and applications in your space. Let's focus a bit more now on your interests in field research and what's happening in the environmental side. In the past few years, you've been involved in a number of high-profile investigations concerning infectious disease outbreaks, such as invasive group A strep in the Southwest and the crypto emergence in the Pacific Northwest. What are some of the environmental drivers of these unusual occurrences? What have you learned that might be useful for predicting and possibly minimizing future migrating pathogen outbreaks?
Yeah, thanks for that question, Rich. And this is an area that I'm totally fascinated by, is really the exploration of where some of these pathogens come from, how they move around, how old they are, why they come into new areas, and what are the drivers. And so you mentioned Cryptococcus in the Pacific Northwest. This is a really good case example where, without diving deep in really a multidisciplinary and One Health way, we sometimes make really kind of wild assumptions about what may cause an outbreak or an emergence of a disease. So Cryptococcus gattii, we'll start there. Cryptococcus gattii, it's one of the Cryptococcus fungal species that can cause infections in humans. It's actually turned out to be a complex of several species, but we'll just call it gattii. So it has normally been found in tropical and subtropical parts of the world. We've known this, the Amazon, some tropical areas in Africa, where this fungus seems to survive and thrive in decaying trees, decaying vegetation, tree holes, those types of things. And then people can get exposed and get sick, and then either have a very serious pulmonary disease, or it gets out of the lungs and actually gets into the CSF and causes meningitis or encephalitis. With Cryptococcus neoformans, which a lot of people know about, which is, we know we can spread in or get spread in pigeon droppings, and then people can breathe it in. Therefore, it's found around the world. Gattii does seem to be specific towards kind of the decaying vegetation. So, which is why it's going to be common in tropical areas where there's just a lot more vegetation and dying and regrowth and all of that. Well, it's very surprising that in about the turn of the millennium, in about 2000, that some cases were showing up in the Pacific Northwest, namely on Vancouver Island and some coastal areas. And then people's pets got sick, and then they started looking closer, and they found more cases and more examples in wildlife, and even including in some of the marine mammals that are off the coast of between Vancouver Island and mainland Vancouver. Then in a couple of more years, this was starting to be seen in Washington State, because our radar is up. We're looking for more of these cases, as well as in certain areas in certain parts of Oregon. So this is very unusual. We didn't expect this, and people are wondering, how did this happen? I think, in today's day and age, the automatic boogeyman for these types of things is we just got to say global warming. But that doesn't tell you the mechanism for how something might transfer up to an area where you've never seen it before. It might change a local environment that allows for some microhabitats for pathogens that are microbes that didn't exist before. But we didn't really know how it got up there. And did it just happen in 2000 or 1999 when the first case was found? So the CDC asked us if we could do some analysis of this interesting situation, do some genomic sequencing, because we were with TGen, we had been really kind of leading in microbial genomics for public health. And so it was an area we jumped in. There wasn't a whole lot that had been done in this space. So we sequenced, identified there were some very specific clones of this fungus up in the Pacific Northwest. But interestingly enough, is we also saw that even though they were clones, everybody else's tools, they looked really identical. We could actually see some evolution that had been occurring, because we could look at every single mutation in the genome and give an assessment of not only how much evolution is occurring, how old is it? And by doing that, we could actually put a molecular clock on it, and we were able to determine that this actually didn't just happen in around 2000, but it wasn't there for a very long time, probably no more than 100 years, maybe 70 to 90 years that this had occurred up in the Pacific Northwest. So that gives us something to actually bring into our investigation and gather more intel. What happened? How did he get up there? So we started sequencing a lot of Cryptococcus gattii strains from around the world, especially in the Amazon region, where we determined the isolates or the clones that moved up to the Pacific Northwest absolutely had to have come out of Brazil. That's the most closely related we could find. In fact, we found a couple of strains that were really closely related. So most likely on the eastern side of Brazil, a port city of Recife. That's where we found the most closely related. So that tells us, okay, how does a fungus get from a port city in Brazil up to the Pacific Northwest? Well, things didn't travel very easily 100 years ago across the Western Hemisphere, from the eastern side up to the Pacific Northwest. And sailing ships would have had to bring it all the way down around the southern horn of South America and then back up the Pacific coast side. And so while there was some of that, we didn't see a whole lot. But what also happened right about this time, and for me this is fascinating, is that of course, what I call the Teddy Roosevelt effect, the Panama Canal, that opened up the seaways on both sides of the hemisphere. And now ships from Brazil or the East Coast could easily passage through and get up to the Pacific Northwest and take materials up there. And so that still doesn't give us necessarily the answer of how it or what was a true mechanism, but it does seem like ships could have brought something up there. We could not find good information about what would have been contaminated with the fungus, other than sailing ships back then and ships now. They all take on ballast water when they're in port, and then they dump out ballast water also when they're in port. So they transmit a lot of things that are in the water, including microbes and as well as crustaceans and even small fish, by doing this. So that's how things move around the planet a lot of times. And we also know that this fungus survives well in the ocean water through some other studies that were being done. And one more piece of evidence, the fact that there were sea mammals, multiple porpoises and dolphins getting sick with this fungus, told us that the fungus was absolutely in the water up in the Pacific Northwest. So the thought is maybe ships took it 100 years ago or 90 years ago when the Panama Canal opened and contaminated the harbors and the coastal line. One more fascinating question, though, is how on earth, if that's what happened, how did the landscape get contaminated with this fungus for it to then grow within the Pacific Northwest region? Well, one hint is that we looked where it was found in every location. It was really near the water. It was on the coastal lands, maybe as far up as a few hundred meters from the coast, but it wasn't deep out in the forest or hadn't been found. So it's quite possible that it moved from the water up into the forest. And one incident happened about 50 years ago, 55 years ago. Now almost 60 years ago, I'm dating myself, was the Great Alaskan Earthquake of 1964 caused massive tsunamis. And this was the second largest earthquake in known history, and caused such massive tsunamis up and down the coast that it would push massive amounts of water up into the forested regions in the Pacific Northwest, and absolutely did, and could have taken the fungus up there. And now the fungus is in the forest. There's decaying wood material that you have, and then it could set up shop. So it's really trying to take this multidisciplinary view, this One Health view, bring in intel both from the genomics as well as everything else that we could gather to help piece together, I think, a pretty good story anyways, about how pathogen can move from one part of the world to another part of the world and what the mechanisms of that. And every time we see a new pathogen showing up, there's probably, maybe multiple causes, but there's definitely an interesting story behind that. And that kind of historic epidemiologic work is totally fascinating to me.
Yeah, Dave, that's an amazing story you tell there, and it brings to mind articles that appear once in a while in the popular press, concerns about the thawing tundra, and what do we not know? Are pathogens being released? Are there viruses there that we should be worried about? What are your thoughts? Have you looked much into that area?
Yeah, so I have in a couple different ways, and I think it's a very interesting idea. Let me just step back for a little bit further back in history. This is going to sound a little crazy, but if we go 200 million years ago, we had Pangea, and Pangea was breaking up into these multiple continents. Well, these continents were connected, and they were in different locations, and they had different ecologies. So like one of the great forested chunks of land in the more southern part of the globe broke off and actually moved down and became Antarctica. Now, Antarctica is completely frozen wasteland, but I shouldn't say a wasteland, but frozen in most places. But underneath that is not just rock. There's old soil of where there was enormity of plant life and animal life, but certainly other microbes that could be pathogens are underneath that frozen ice that now we're starting to drill into and get cores out of and doing more metagenomic analysis to see what may have been growing on that land way before it became what's called Antarctica now. So to me, that's interesting, is that we know that there are microbes that are covered up, and whether or not it's from warming in certain areas or from humans going in and purposely exploring, it is most likely we will find things that that we've never been exposed to. Or if we did, say up in the northern tundra areas, have been covered over for a long period of time as we dig up old remains of prehistoric humans and others. They absolutely had their own microbes that they were carrying and would have died with. We may be able to identify those, hopefully not exposing ourselves to those, but people have gone and dug up cadavers from 1918 flu pandemic that were covered in tundra to see if we can get live virus out of it. Yeah, I think it's a possibility, and I think it's not a it's a great likelihood that we will discover some pathogens this way. Will it cause a pandemic? I don't know. I don't. I have no under expectation of the human ability to accidentally discover something and spread something.
Well put. Yeah, again, Dave, thank you. Fascinating discussions. Are there any recent studies that where you find examples of the types of outbreaks that are becoming more commonplace? And if so, what are the takeaways?
Yeah, so let's maybe we can just talk a little bit about the pandemic here for a second. You guys have probably talked forever about the pandemic, but the pandemic is a virus that we actually know more about this virus than probably any other pathogen ever studied, just because the entire world was focused on it for several years. Uh, we still don't know exactly where it came from and how it spread, but it's become the most common virus or pathogen now found, I should say pathogen now found in the world. Everybody was exposed to it. Almost everybody got sick from it. And this virus has evolved in a way to make it act like the very commonplace coronaviruses that we have, that it's very easily transmitted. It's an upper respiratory virus, and it is adapted strategies to be able to survive and thrive in a human environment. And probably the biggest adaptation there is the fact that it can have this kind of fluctuating mutations in the receptor binding domain of the spike protein where antibodies would bind to. And it just only needs to make a couple of mutations for those antibodies to be less effective, and then allow for more spread in a population to occur. And then you get this kind of cycling happening. And that's exactly what happens with other human coronaviruses. So this has become another human coronavirus. It's most likely that the other ones that we had started out as pandemics like this. They spill over from rodents or from bats or from some other animal, and it took a little bit of time for it to adapt, probably longer than this one did, to adapt to the human species, but it's become this very effective strategy for survival. So we see that some things will spill over or potentially leak out of a lab or whatever the driving cause was of this last pandemic. And then but they end up finding a strategy that other pathogens have already found. If not, then it becomes extinct and it goes away, or it becomes something that just never causes really big problems. So other coronaviruses like MERS, the Middle Eastern Respiratory Syndrome virus, that spills over from camels to humans somewhat frequently, and human to human transmission can occur, but it just not becoming adapted to humans, and therefore is not a problem. But oftentimes these pathogens do.
I recall that there was the so-called Russian flu in the late 1880s and early 1890s that many people speculated one of the 4 coronaviruses that now are part of the common cold may have caused that because the symptoms look like coronavirus. What do you think of that?
I think the timing is pretty remarkable. I think there's some evidence to suggest that really was another coronavirus pandemic, but there was no testing back then, so couldn't tell the difference between a coronavirus or a influenza virus. In fact, even understand what that a virus would have been causing it. So I do think that is a possibility, and I do think that a lot of times we know that a lot of the organisms that we get, the microbes that we get that are pathogenic to us, and that we can spread around, do come into us from nature or from animals. In fact, probably most do, and did over some period of time. So again, really fascinating. We look at something like tuberculosis, which it's still, I think, the most important infectious disease in the world. 1 person every 20 seconds dies from tuberculosis. We cannot forget about this scourge that affects almost everywhere but the US, and even some parts of Europe, except we do get cases because of migration and travel. But it is incredibly important, and it's become really adapted to humans, but it wasn't always a human species. It most likely came into us from either animals that we domesticated or from wildlife. But really, when we look at the old history of it, we know that it spread at the same time that humans were actually settling down into what we would say more modern, more civilization, rather and less kind of hunter-gatherer lifestyle. Really only about 6,000 years ago, we're domesticating animals, we're living closer to our animals, and we're sharing a lot of microbes with them. And that's likely, TB is not very old. It's probably only the several thousand years old kind of category. And so we know that humans have this profound effect of not only moving microbes around the world, but causing evolution of those microbes so that they actually become something that's adapted to us. So maybe this gets to your question a bit better, Rich, is that we are seeing that outbreaks are becoming, in depends on the type of microbe, but they're becoming larger and can affect parts of the planet that in past they wouldn't. We saw the Ebola outbreak in West Africa explode because for the first time it's showing up really in in metropolitan regions and where people are densely packed in ways that where we normally would see Ebola in sub-Saharan Africa and in the Congo region and other places, that it would really be pretty limited to the villages that were affected and could spread a little bit. But here's this explosion, and then we're seeing Ebola actually show up in the Western Hemisphere because of plane travel. And of course, it didn't go on to cause a massive epidemic or massive pandemic, but it definitely exploded in a way that it wouldn't have, 100 years ago. We just move things around so fast. Up until this pandemic, the fastest moving virus ever in the history of the world would have been the 2009 flu pandemic. And that is because of the mobility of humans and the ease of transmission of influenza. We've never seen anything move that fast, but you have a new virus of which everybody is susceptible, and therefore it just was able to move really quickly as we moved around the globe in a way that we knew was happening, but we hadn't seen before. And now with this pandemic, we get a new variant of an existing virus of which there's still a lot of immunity that's been circulating. in South Africa in, around Thanksgiving, and then boom, the whole world gets Omicron within about a month. It's amazing how fast we humans can move microbes around the world, especially if those microbes are really well adapted to transmitting between us.
They just love us. We're their transport medium. It's fantastic for them.
Yeah.
Yeah, fascinating discussion. I think it's a brilliant tee up to the next question on dengue. For us, it is the perfect poster child, teeing up the mobilization of the diagnostics community. It's been out there. It's been in countries that have been recipients of lower, less amounts of funding. CDC's been pushing for a long time to get better dengue tests out there, but it just hasn't gotten a lot of traction because it's seasonal, it's regional, it hasn't impacted high income regions of the world too much yet. And now suddenly WHO sends out this warning. And it's echoed somewhat by CDC. And what is it portending for us? How serious should we take this? Is that an indicator of a whole new category of diseases that are likely to start pushing up into southern United States, up the seaboard? Will it spread through, blood donations, or is it going to be, just what's next?
Yeah, it's a great question. And yeah, I think dengue is it's a really interesting case, and it's a good poster child for a few different aspects of infectious diseases, epidemiology, and movement and risk of pathogens moving around the world. So, okay, dengue is a virus, flavivirus, spread by mosquitoes, right? And it's very specific mosquitoes typically that need to be able to spread this virus. And the virus is really just a human adapted virus. And so you need infected humans, and you need the vector, and you need the right environment then for people to get exposed to that vector, and have the transmission cycle going, as opposed to say something like a West Nile virus. When that came in to the US back in 1999, up in the Bronx, New York area, and then quickly spread across the country in a matter of a few years, really affecting bird populations, and then causing a lot of human disease, and still causing a lot of human disease today, in the US as well as in many places around the world. That virus actually is really more of a mosquito bird virus, and then humans are the accidental host. And therefore it was able to set up shop where you had a large number of vectors could spread this, a large number of birds could be infected, and therefore it can maintain itself in an environment. And then people get exposed when they get exposed to really to mosquitoes in many parts of the world. Or many parts of the US. So West Nile is going to be one of those things that it just needed to show up in the US, and then it took off.
And of course, you had horses as a reservoir, right, to help continue propagating and storing that virus, if you will.
Yeah, so horses actually end up being more of a dead end host as well. They can amplify a fair amount of virus, unlike humans, but the birds is the really important piece there. But if you think about with dengue being a human virus, you don't need, you don't have then the environmental driver, unless you have infected humans, right? So you need to have infected humans as well as then the specific vector mosquitoes. So I have just a little bit of background on this. Just thinking about even down in Arizona, hot, dry, people think of it as that's the land of valley fever, and things that are really thought of to be in the desert Southwest, and not typically vector-borne disease or mosquito-borne disease. Well, back in the 90s, when I was working there at the state health department for Arizona, I was able to publish on the reemergence of Aedes aegypti mosquitoes in Arizona for the first time in 50 years. So we had massive, really good, but probably over-toxicating vector control programs in the US for quite some time, but it wiped out a lot of mosquito vectors from most parts of the US. Well, and so we likely had Aedes aegypti going back, and we know we did back 50, 60 years ago. But then all of a sudden it starts to show up in a couple neighborhoods, say in the Tucson region, then in the Phoenix region. And now you fast forward, we can find Aedes aegypti throughout the entirety of the metropolitan regions of the the Southwest, as well as in many parts of California, seen in New Mexico, and certainly Texas and Florida, and also Aedes albopictus. These are the two good vectors. So now you have the vector, but you don't really have much of a human population. We have migration happening. We have travel, people coming up from Mexico and Latin America, where you do have dengue circulating. Now it provides an opportunity for a mosquito to accidentally find the right person with the virus to get infected and then go infect somebody else. And that threat has always been there. But for the very first time last year, a human case showed up in Maricopa County. And then, of course, they do a really good analysis of the neighborhood where that occurred. Maricopa County is where Phoenix is. They identified multiple neighbors in that area that had also been exposed by doing some blood serology testing. So there had been some local circulation. So now we know that we're getting the virus circulating because there's enough people that have come up, like I said, either through travel or immigration into the state from Latin America that would be bringing the virus with them. The same thing has happened now in Southern California for the first time this year. Dengue is now shown up to be spread locally, a couple different instances in the LA and Long Beach region. So this is starting to happen, and it's not going to stop, because the vectors are really well situated now in these environments that they moved into. And we have so much migration of humans that we're just going to see more and more of this. So this is kind of more of like a slow roll of this. It's not taking over certainly like COVID did or like West Nile did, but it is like watching the kind of that slow moving tsunami coming up, and we know it's just going to keep rolling, and we're not going to be able to stop that. So the best thing that we can do is really having good public health preventative measures in areas where we know there's risk, and the risk is where this vector is, and then where we have movement of people that or travel from areas where dengue was. So we can do some good preventative measures, but we're not gonna hold the ocean back here.
Yeah. It's very interesting just to think about. We talked to a public health officer for Solano County, Southern counties of north of San Francisco. And he's concerned about what to do with all of this. What do you do with sequencing? What do you do with wastewater testing? How does it actually inform public health policy? How does it form budget allocations? Yeah. What do you even do for budget requests? And because it truly is population wide, but doesn't necessarily point to a course of action. That's always in the back of my mind as well here. For West Nile, for SARS-CoV-2, there were calls to action, to your point, right? It's out there, it's fought, the migratory pathways, whatever it's, where it's headed. But with dengue, what do you do from a public health perspective?
So Dave, given a situation like dengue and others that are likely to occur over the next many years, how could a diagnostics industry complement and build from your studies and those of others about these new pathogen outbreaks? For instance, are better surveillance tools required? And if they are, how would they be used? Are we missing key diagnostic tests? What do you think about that?
Yeah, these are really good questions. I think, and I was listening to one of your earlier conversations, probably with this public health officer, and I heard something that I've heard in other circles, which is, how do we use this kind of fancy new technology to really make a difference in my particular world? And maybe that's in public health, or maybe it's in clinical diagnostics. And again, I go back to, we're really only limited by our imagination as well as our understanding of the capabilities and limitations of this technology. So with something like with dengue, let me just talk to how we're using West Nile and using pathogen intelligence with West Nile, and maybe we can find the way to relate this with dengue. We know West Nile's around us, but it's not good enough to just say, oh, well, there's a lot of West Nile, so we shouldn't do any testing, or we know it's out there in the environment, why would we look? Working very closely with an entity like ours, a research institute like ours, working with the local vector control agency and public health department, we they've mapped out, and they do lots of great mosquito capturing. They even do testing and look for the presence of West Nile, and they can find it in different areas. Well, that's just detection. That is maybe one step better than what, kind of the mainstay of public health is, which is just to essentially sit back and gather case data that's sent in and say, oh, it's increasing here, it's decreasing there. But when we're starting to now, we're getting the sample, we can actually go in and do the genomic analysis and actually look at within these positive mosquito pools that are being tested, how much virus, how much variability of the virus. That tells us if transmission is happening at that location versus another location where maybe one location you have no variability. There's no viral diversity. There's probably just some localized transmission happening. But in other areas where you see a lot of viral diversity, and you also can be testing for looking at blood meal analysis. We can look at the different species of birds that are being fed on by these mosquitoes. We can understand, is that site more of a hub for the virus, more of a source that goes out to other areas versus sinks? If so, now you can focus your public health energy and your public health efforts. This is precision epidemiology in a One Health space to really go in and say, we put our efforts here, we're going to knock out 90% of where the virus is coming from and say this 1 square mile or this 10 square miles. That is not just a hypothetical. We are absolutely seeing that. So we've got great researchers at TGen here that are enmeshed and working with public health, so they understand it, they understand how to use it, the knowledge from it, and we can build systems together so that we can really improve public health and the utility of these technologies. So thinking about with dengue, we may be seeing some very similar things. Improve surveillance by monitoring what's actually, are we seeing multiple variants of this virus, or are we really just seeing one introduction at a time, which is most likely what we're going to see for a while, because that's, we're going to have introductions coming up from the south. But we can actually get intel by further exploring the genetic material of the samples that we're gathering. And then this, looking at diagnostics, we can have very precise diagnostics that do tell us not is dengue present, but also which of the 4 major types of dengue is present. Because we do know that you get dengue once, you could dengue fever, not fun, breakbone fever, but if you get it again with another variant, you're at much higher risk now for dengue hemorrhagic fever, a much more serious, deadly disease. And so we could actually be looking and monitoring when places are getting more and more dengue, what were you exposed to? What is the risk of having a much more serious illness? We're also using that, the genomic information, I think, is really going to help us identify better ways to both treat patients. Obviously, antivirals are becoming more precise, and that'll be useful. But also, I think for how we respond in the environment, I got to mention one other thing. We have one of our researchers here is working with some academics at University of Arizona and other places where they could see some of the natural viruses that mosquitoes have are inhibitory towards some of the arboviruses, or the ones that are serious to us. So we could almost think about inhibitory viral vaccines for mosquitoes. There's lots of really cool things happening in the vector control space, as we've seen, releasing mosquitoes that can't reproduce, or ones with other vaccines. All of this is happening because we're able to gather this knowledge. And then in the diagnostic space, diagnostics are moving towards, just like with sequencing, more rapid, more informative, easier to use, cheaper, home-based, field-based, all while maintaining the great characteristics we're getting from molecular and genomics approaches. I want to mention one example. We're working with some researchers in South Africa, looking at wildlife in South Africa that for a number of pathogens, but in this case, looking at Mycobacterium tuberculosis as well as Mycobacterium bovis in elephants and in water buffalo and others. What they need is a very accurate test, and we don't have that with just some of the existing lateral flow tests or things that they could do in the field. They have to take samples, go back, run PCR tests to say, oh, you know what, we got to now go back and treat this elephant for TB so it doesn't transmit more to other animals in a wildlife park. We're actually taking that technology and making it very field-based, where you can run kind of what we call isothermic reactions. So PCR, without having to use a thermocycler in the field, you actually can just make the reaction happen in your hand, holding it, and then run that out on a lateral flow assay or something else in the field that tells you now genomically it's this strain versus this strain. And that makes all the difference for how you can respond in the field. I think we're going to be able to take those kind of technologies, see more and more of that happening for things in our own backyard.
By the way, we have been trying like crazy to get the diagnostics industry in the United States to work on dengue products and to make sure they include the 4 serotypes, as you are suggesting. It's just absolutely critical, just as you say.
Yeah, and I mentioned these 4 serotypes of dengue and being able to look then for them, and that makes a real difference on how we're going to treat patients. The same is true for almost any of these microbes. The better resolution we have, the more we can differentiate between what we thought were maybe just different strains, but they end up being different species, and they may have different phenotypic effects, but our current diagnostics only say, hey, it's this. So like, for instance, Cryptococcus fungus, you actually send it to lab, you just get a Cryptococcus species positive back by and large. And now, your physician is thinking, well, it's probably neoformans, probably not, probably this is probably going to be an immune suppressed individual. I need to treat it that way, which is where most neoformans is. But we're seeing that if you can differentiate and see Gaddii, you may be at a higher risk for getting a neurologic disease from this. And it may be people that are not immune suppressed, which happens more often with Gaddii. So we need to have the diagnostics that actually separate out these species. And as I mentioned, as we looked at Gaddii more and more, all the different, what we just called major molecular types, those turn out to be actually different species of fungi. And we're just starting to learn what might be the phenotypic differences. So our knowledge base is growing daily, and we're learning more about pathogens that we thought maybe were all one species, but actually were turning out to be several different ones. And this is not just a lumpers and splitters argument about species. This is actually different things that we just lumped together because we didn't have the resolution to pull them apart before. And maybe we can have more precise medicine responding to the actual infection a person has, not just the type of infection a person has.
Yeah, that's very interesting. So, Dave, you talked a little bit about pathogen intelligence, and I know you're interested in this whole area of epidemiology and pathogen intelligence. Tell us a little bit about your efforts in that area.
Yeah, as you reference, epidemiology and pathogen intelligence, bringing those together is giving us a lot of power in really advancing public health, but also clinical medicine. So a couple of examples. We've applied this, and as I mentioned, really to cool stories and trying to understand how diseases spread around, but also really critically in disease outbreaks and helping to find a source to help stop an outbreak or to prove whether or not an outbreak exists. So we've actually been involved in very specific and important disease outbreaks by bringing genomics in and building this intel and combining it with epidemiology. One case example is, you may recall, probably, what was it, about 10 years ago, we had a compounding pharmacy in New England that was making back pain medication as a steroid, methylprednisolone, but they were doing it illegally as a compounding pharmacy and sending it across the country without prescription. So they're breaking all sorts of laws doing it, but it was also, turns out, we find later on, highly contaminated, gross facilities. This ended up being the largest medical product contamination case in US history. 76 people died of fungal meningitis, almost 1,000 cases of fungal meningitis around the country. And we were able to, working with CDC and the FDA, we were able to test samples that were coming from all these different states, coming out of the vials as well as out of the patients, and showed they all had the exact same strain of this fungus, Xerohilum, that would just normally be a mold type fungus you see growing on plants. But it was also found then in the compounding pharmacy facility. And so it was a real lock. But what was interesting is this was a criminal investigation. I had to testify in a federal murder trial with this information. And this really is a forensic epi use of pathogen intelligence to nail this down. Now, it seems pretty obvious in hindsight, but we really could only say, oh yeah, it all looks like Xerohilum, so therefore it all must be part of the outbreak. By using the genomics, it was an absolute lock, and there was convictions across the board and shut down that facility, which it absolutely needed to be. We now use that on a very local level too. We'll have a hospital come to us. So we had a hospital in the Southwest come to us and say, hey, we have a surgeon that we know was colonized with MRSA, because a lot of people, we have MRSA in our nose, the methicillin-resistant Staph aureus, and several patients that was with that surgeon ended up becoming infected with MRSA post-surgery, and 2 of them died. We need to know, did the surgeon cause this outbreak? Because there's a whole lot of ramifications, as you can imagine, around that, for the surgeon, for the hospital, obviously for the patients. So by working with the hospital epidemiologists and the infection control staff, we're able to obtain samples from those patients, from the surgeon, from other patients in the hospital, and from the community. By doing all the analysis, the sequencing analysis, we were able to show that not only was the surgeon strain different from the patients, all the patients had different strains as well. This means this is more likely what happens. Instead of a nosocomial outbreak or a healthcare caused outbreak, people are bringing their own pathogens with them. We're not testing ahead of time. And then they auto-infect after surgery or after treatment. And then it looks like all of a sudden we have a hospital outbreak. So the findings of that, obviously enormous healthcare savings in that the hospital wasn't liable and the physician wasn't liable. It doesn't really help out the patients, other than we can do better for our patients and do better screening ahead of time, better surveillance ahead of time, to try to prevent these types of outbreaks from occurring. So you can see how we can use this in different, this intelligence along with epidemiology, whether it's in the hospital setting or in the government public health setting, to really come together and solve these problems. One last thing I want to mention, though, is that's still somewhat reactionary. What we want to do is be very proactive now. As I mentioned, sequencing is becoming very easy to make routine. It's less expensive. We know how to handle the data. We can do automated analysis. We can produce reports that others can use without having to be bioinformaticists. So why are we really transforming our surveillance systems to really include genomics? So we have that information ahead of time. So we started, this is before the pandemic, we started one of the first genomic surveillance systems, statewide surveillance systems in the country, in this case, looking for invasive group A strep and look for outbreaks that may be caused by this. And because we had done some in other outbreaks associated with group A strep, we just had built up some knowledge base. So we got the state health department passed a public health rule. All strains of invasive group A strep had to be sent in for sequencing. And then we would receive them, sequence them, build the different trees for all the different types of group A strep that are out there. And then we could actually see the very first cases of clusters. We could see that when you have 2 cases that had the exact same strain, no mutations between them, there's definitely a transmission event. Maybe there's a third party in there too, but it's very localized. And if you see that, you can now be precise with your epi and resources and go in and investigate that and say, hey, what are the commonalities? Are we about to have something larger? How can we stop this from spreading? As opposed to the way it is now, public health is gathering in data, the number of cases that a hospital has to report or was seen in the lab. There's nothing connecting, and you either don't do anything, because you can't do anything with that information other than to say, hey, it looks like we're having an increase, or no, it looks like the same as last year. You can actually say, hey, here is where you could focus your limited time and ability and resources, focus in on this and stop this. And so we've seen that now. We've seen some outbreaks that have grown really large because they weren't jumped on the first 2 cases, because we're still learning how best to incorporate this into the public health response. But I think this has so much power in public health as well as in medicine to not only have significant healthcare cost savings, but really reduce morbidity and mortality and better use our limited public health resources. So it's not a matter of, hey, this is fancy and new and therefore maybe we should use it. It's here's something that's actually going to revolutionize and transform public health if we know how to use it and we have the imagination to put it into place.
It occurs to me, Dave, that perhaps, looking for a catalyzing or an enriching event could then allow those tools, the genomic, surveillance, to be used even more effectively. Several years ago, we had, the opportunity to work with International Medical Corps, and they were wondering, this was before the advent of many of the tools you're talking about, wondering about the possibility of putting together a diagnostics field kit. It's ready to go. There's a natural disaster, there's a flood, there's a tornado, there's a hurricane. There's an opportunity for lots of people to come together, lots of disruption of the local flora and fauna. And, can they be better prepared instead of waiting for that cholera outbreak, waiting for, what they are probably is going to happen? They could have the better surveillance tool and then bring in the supplies that are needed ahead of time.
Actually design it properly, right? Based on this. Is it a local event we have to just surround, or do we have to treat it as though it's more of an epidemic?
Yeah. What I think one of those toolkits, and I think it's a great idea, and I've certainly been involved in a number of public health responses, both locally and globally. When I was in public health, and I was the state epidemiologist for Arizona for a while, and one of the interesting things we had to respond to was Hurricane Katrina, even in Arizona, and that's because we had a lot of people coming to us. We set up at one of those very large shelters, and this was actually a lot of the sickest of the sickest that were in the dome in New Orleans came to Arizona. So we had to set that up and know full well that a lot of people were exposed to things we had no idea what to look for, that were really in a bind that way. But if you're actually on the ground in New Orleans and maybe you're testing all that brackish water that people had to walk through and get exposed to, and you're using tools like metagenomics where you're able to just take samples and just sequence everything there and look for things that people are being exposed to, you would have been able to potentially identify the Vibrio vulnificans, which was in there and then ended up causing a lot of infections, especially through cuts and scrapes and some serious infections as well. But that's one of those kind of flesh-eating diseases, and without even knowing that you needed to look for that. So, I think that there's these great unbiased tools now too that the genomic age has brought us to. Molecular testing, as I mentioned, we're very focused. We're looking for very specific things. And we've done that with sequencing as well. In some regards, we're either we need to get an isolate and then we have to sequence that out, which limits us to being able to test samples, or we test a clinical sample, but we're looking for some very targeted information. Now, with metagenomic sequencing, we're actually just look at all the genomic material there and then look for anything that's of interest. And that's becoming more and more commonplace, used for exploring, like, people with a disease of a pathogen of unknown origin. Maybe it's a neurologic disease, like when we had this enterovirus D68 that was causing polio-like disease in children. We were able to apply metagenomics to samples to look for the presence of anything that may be causing this and finding signal of one particular enterovirus that could lead to a polio-like illness. Or in the environment, looking for the presence of things that we wouldn't know to look for. So it's very unbiased look where we can just now handle all that data. And the tools, even though we could do this 10 years ago, the tools now, the bioinformatic tools, are really allowing this to be put into place. So I think field-based sequencing and genomics like that is actually going to be really useful to help us kind of focus in our efforts to respond to emergencies like you just suggested.
Yeah. So with all these disease outbreaks that are occurring now and likely to continue, we know that the sentinels in public health are often really astute physicians in the community who see something, say, I better report that. There's something weird going on here. And I just wonder, it's often the case with these newer things, these physicians have probably never seen anything like this before. They don't know the symptoms, or they think it's something else that's more common. Just wondering, is there a need for helping to train people more broadly, or is this an opportunity for AI actually to come to the assistance of physicians to identify things that they wouldn't normally think of?
Yeah, that's a great question. Having been in the infectious disease epi world for 30 years, the single greatest tool of an epidemiologist for identifying something is the astute clinician. Uh, but that is a very random, haphazard way to detect and respond to disease outbreaks. We still need that astute clinician, but we need to give them better tools to be looking for things that are, that's weird. Oh, wait, we're not seeing that. I've never seen this show up before. Because that process happens, but a lot of things fall through the cracks. And sometimes it takes, it could take days to weeks to months to really determine what's going on. So by the fact that we have this genomic revolution happening, what that really means is that this is kind of going back to the beginning of our discussion, is that we're digitizing the biological world, right? We're turning the A's, G's, T's, and C's into ones and zeros. And that means we can have a whole host of tools that can actually help parse that out and really make sense out of that data, turn it into information, and then help us build knowledge. And that means that the advancement of these tools and the advancement of the way that we approach data science is also going to advance how we're able to apply genomics. And so artificial intelligence, this has been something that's been building in the background and it's just exploded in the last few years. But, just the idea of even machine learning, where we're actually using tools that learn from its last data set to give it more context and knowledge for the next analysis, that's been going on for a while, and that's helped us better parse out and understand what might be going on, either with these very complex metagenomic data sets, or to help us understand and build things like phylogenetic trees. So we've been using kind of that early stages of artificial intelligence, but now that we're moving into large language models, we're actually able to take something like a data science and make it like human discussion. And in a way, and at whatever level you're at. So at a clinical level, really transforming all of this into things that are useful for a clinician, whether that's identifying some kind of rare occurrence of something and say, hey, we've never seen this in a human sample before. Maybe you should look closely at this, to actually looking for trends that are just occurring in the background, and just the slightest anomalies you wouldn't pick up might actually be leading to something that's much greater. I do believe that there's a lot of ways that AI is really going to continue to help us. In fact, it will be what continues to revolutionize this, because this sequencing is now a data science. It's become so easy to transform all of that into data. And now it's going to be these new tools. And we think about large language models and how it parses out human language, but of course, there's that natural understanding that we're talking about the language of DNA here now, and how we can better parse that out and understand what even slightest changes might mean, not only for this patient, but maybe for this organism 5 years from now, as we're looking at samples from across the world all at the same time. So I think this is very much we need Imagineers here to help us figure out where this can take us, and then build kind of strategies around there to get there.
Very exciting. Very, very exciting. I think that's the end of our questions. That was a lot of fun.
That was a very nice discussion.
Yeah. Dave, thanks so much for joining us. It was a great pleasure to talk with you, and I think we and our listeners will have learned a lot from this.
Yeah, thank you, Mickey and Rich. Those were great questions, and it was a lot of fun discussing this, and just even visioning more about the future, and appreciate what you guys do and helping to actually make some of that a reality.
Couldn't agree more, Dave. Thank you very much.
Thank you.
Halteres Presents is produced by Halteres Associates, a US-based bioscience consultancy that provides strategic and tactical services in the areas of diagnostics, medical devices, and life science research to clients of all sizes. The views, information, or opinions expressed during the episode are solely those of the individuals involved, and Halteres Associates is not responsible for any errors or omissions or for the results obtained from the use of this information. The information provided in this episode is for informational or educational purposes only and is not intended or implied to be a substitute for professional medical advice, diagnosis, or treatment. Halteres Associates would like to say thank you to this episode's guest or guests, and thank you for listening to this episode of Halteres Presents.